Okay, good morning, everyone. This is Ioannis from Flojo, and today I'm going to be talking about the high-dimensional data analysis with Flojo version 10. If you have any questions, please make sure to type them in the chat box or in the Q&A box, and I'll make sure to address them on the fly. So we're gonna be talking in this webinar. I divided in 3 parts. So in the first part, I'm gonna introduce you to the Flojo plugins. So those third-party algorithms that we can use and install in Flojo for free to analyze high-dimensional data. And then I'm gonna show you the background of the high-dimensional workflow, when to use it, how to use this workflow, and how to adapt it to your experiments. And in the last part, we're gonna go into a live demo. So we're gonna use some samples, some multi-parameter flow samples, and see how we can apply all of those tools into this high-dimensional workflow. So let's get started here with a bit of basics. Basically, flow cytometry has evolved from low flow experiments, so using only few antibodies, and in Flojo we can analyze those data quite easily. So we can overlay the populations, we can check in the end-by-end view, and with manual gating in most of the cases, it's rather easy to identify the different cell subsets within our datasets. But of course, flow cytometry has evolved, and now we have the need to identify even further immune cell types, and even more information about rare populations and so forth. And now it all becomes slightly more complicated. Yeah, so you can appreciate here the different combination of parameters that we can have compared to low parameter flow experiments. So now it becomes quite more difficult to gauge on each individual cell population that we have in our data set. Therefore, we're going to apply a different workflow so that we don't lose any information in our data set. And this is where exactly the high-dimensional workflow comes in place. So now we can analyze those data and generate a 2-dimensional space. Yeah, so there we can visualize all of the different cell population at once. Yeah, so instead now of gating on many different 2D plots, now we can use with dimensionality reduction 1 2D plot, where each one of our phenotypes will be embedded in that space. And then we can use additional tools to gate and to extract all the information we need. So let me get started with the plugins. So plugins are algorithms that help us analyze those high-dimensional data. So there are many different algorithms that you can find in the Flojo Exchange, and I'm just gonna go quickly in our website here to show you where you can find and download those. So here I am at the Flojo Exchange at the Flojo website, flojo.com, and under the Flojo Exchange here, you will find a list of many different plugins. So don't get overwhelmed there. There's a lot of plugins, but simply because there's many different experiments that you can perform out there, there's different reasons to use each one of those. So all of those you can download for free. We always update it to the latest trends in cytometry. So yeah, you will also, if you sign up in our newsletter, you will get an email when we have something new out there. So when you want to download one of those plugins, you can simply here filter for Flojo, and you can, for example, select here the different kinds of categories. I can select quality control, where I can, for example, download Flow AI. So Flow AI is a quality control plugin that finds anomalies in flow cytometric data. And I can click here and I can download this plugin for free. Otherwise, we also have another solution, which is the plugin play bundle. So basically the bundle is a zip file. So download a zip file that has multiple different plugins within with the R programming language configured, so you don't need to do anything to download to make those plugins work in your computer. So, but I'll show you here how we usually install each one of the plugins. So then I will press download here, and this will download a zip file, right? So I can use this zip file, I can extract it. Yeah, so this will show me the folder, and within each plugin that you download, you will find two files within. The one is a jar file, .jar file, which is my plugin, and one PDF document, how to flow AI, or how to, each one of the plugins that we have. And there you will find all the instructions of how to make this plugin work and what outputs you get from each plugin. Yeah. In case the plugin uses R to run, the R programming language, you can always find the commands that you need to put here and type in R so that you can make it work. Yeah. And the other file is my jar file, which I need now to copy. Yeah, or yeah, I can copy that. And then I can paste this file into my plugin folder that I have on my computer by a default installation of flows. So for Macs, this is usually in plugins. So this is usually for Macs in the applications and plugins. So it should be right here. So there I can paste all of my jar files, and otherwise I can also have my own folder somewhere in my desktop, for example, and place all my jar files of my plugins. And then the last step is to open Flojo. So we need now to connect Flojo with the destination folder that has all of our plugins, so that it recognizes and we can use them. So, and this you can do under the preference and diagnostics. So in the diagnostics, now here at the bottom, you can see this option, scan for plugins. And you can choose the folder that you have all of your jar files. And after you restart Flojo, you should be able to see under workspace and plugins, a list of all of the plugins that you have. So here there is a long list, but you don't need to have all of them. Yeah. And just to get started, as I mentioned before, we have the Flojo bundle. So under the exchange, you will find this bundle. Yeah, here. So this is basically have all the plugins configured and ready to go. So you don't need to install R or libraries or anything. So it also creates a jar file that you can put in your plugin folder, and then directly we will have 6 of them. Yeah, so You will have flow AI, flow clean, flow sum, cytonorm, index, or environment box. Yeah, just one drag and drop, and then you have 6 of them to get started. So that's a good way to get started with the plugins and use some of them quite easy way. And those, of course, can then be combined with other plugins that you download independently. Okay. Okay, let me go back now to my presentation here. Okay, so the overview of the high dimensional workflow. So I have here some steps that we typically use for our high dimensional workflow. Of course, it doesn't mean that you have to always perform all of them. So I just included all of the steps that we would typically use in an experiment. And we start by normalization. So we have plugins for normalizing, if you have different batches of samples or different, time points, etc. Then the next step is quality control. So we can check for anomalies in our flow cytometric data. Then we have the concatenation, which is basically merging the samples into a single FCS file, no matter what kind of samples we have, treatments or time points, donors, we would merge all of those samples into a simple FCS file. And from now on, from downstream of concatenation, we can perform dimensionality reduction to generate a two-dimensional embedded space, clustering, which will partition our data in phenotypically different populations. And then we can use additional tools to visualize our clusters and our embedded spaces that we have created. So let's get started. Normalization. So we have two different plugins currently for normalization. So we have the cytonorm plugin and the PsyCombine plugin. So when do we use those plugins? So we use those plugins whenever we have different batches of samples. So imagine that you have acquired your samples at day 0 or pre-infection, for example, and then you have a time point day 30 after. Yeah, and there might be some technical variability in your data. So it might be that your antibody lots were different, the cytometer settings slightly different, and that's when we know we have batch effects in our samples, and we can use one or the other plugin. So just an overview here, the cytonorm we must have controls. So we must have controls for each time point. And of course, we must have 2 or more batches or replicates. Yeah. And then cytonorm, based on the controls, it will normalize the signal across the different samples. On the other hand, we have also PsyCombine, which is less restrictive in the use of controls. It works on the concatenated file. So after we merge the samples together, we can perform normalization and remove batch effects. And it works by defining a keyword, a batch ID keyword to set, for example, in that case, we had 3 different batches, 1, 2, 3. We set a keyword for those 3 conditions, and then the PsyCombine will normalize across those different samples. So it performs, it transforms in each batch the values, and then it performs co-clustering to find and revert the expression values to the similar level. So here you can see the differences between the non-corrected and the corrected batches. So that's the two options for normalization. So again, optional, in case your experiment requires it, you can use it. If you can avoid it, it would be great because, it's always a bit scary with normalization that you might remove, some biological information that was simply removed because of this step. And the next topic here in normalization is scaling. So there is no plugin associated with scaling. So I'm just mentioning it as a manual way of adjusting the scaling of our parameters. Yeah. So here I show you two examples of what is the impact of scaling in t-SNE or clustering or any of the downstream analysis. So basically, if we adjust correctly, let's start the opposite. If we don't adjust at all, so here it was a bit of extreme example I took. So I really intentionally scaled incorrectly each one of my parameters and performed t-SNE and clustering. So what I could see in the results was that I do not have any t-SNE islands in my t-SNE anymore. Yeah, so you see we have a bit more of spread of the islands, so we don't have any compact islands anymore. And in some cases, we have overlapping islands here. Yeah, so we need to adjust correctly the scaling and the space, between the positive and the negatives for each one of our parameters that we will use for this. And therefore, then you can see and compare how better our t-SNE and clustering would look like. So here we have more compact and more divided populations. So I can say question here. The question is, cytonorm and cycombine load all the samples to RAM, and in large data sets, this can be a problem as you run out of memory. How would you proceed in this case? Yeah, basically RAM is the most important factor in FlowJo, and especially using large data sets. So what I could suggest here, in case that you run out of memory, so basically your data sets, the size of your data set should be less than your memory, yeah, obviously. But in that case, I would use cycombine. And since, as we mentioned before, you can use your concatenated sample to normalize with cycombine, I would downsample. So you can take a portion of your cells and not your whole data set, merge those samples together, and then use cycombine for normalization. That's the way I would proceed with this. On the cytonorm plugin, so there we use the full sample. So there it would be more difficult to avoid running out of memory. Okay. So let's proceed here. Just what we mentioned here for the scaling, something to be aware that we can. It's good to adjust before continuing all those steps to adjust our scaling for each given parameter. So moving on, we need to perform quality control because there might be during acquisition many different issues that appear in our samples. For example, if we have a clog or an air bubble or any errors throughout the acquisition, this could lead to false discovery or artifacts in our data sets. Yeah, so we have now different plugins that can automatically detect and remove those outliers. Yeah. One example is Flow AI. So it's a very common algorithm to remove anomalies in the data. And it performs 3 statistical tests. It detects changes in the flow rate, instability of the signal acquisition, and also outliers of dynamic range. And all those algorithms, they produce an output of Flow AI good events and bad events. So automatically, it would classify and split any sample that we run those algorithms into good events and bad events. And then we can, additionally, if we want, overlay the population, so we can overlay those good and bad events. So here we see we have in orange some bad events that were removed by the algorithm. And then Flow AI has also the advantage of giving us some visualization. For example, here we can see that during acquisition, the flow rate was decreased. And probably this is the reason why this area at the end of the acquisition was removed. However, there are different plugins for sample cleaning. There is also Flow Clean. So Flow Clean, it does the same thing. It automatically generates populations, but it only performs one statistical test, which is fluorescent anomalies over time. So already you can imagine that Flow Clean is a bit less sensitive than Flow AI because we have 1/3 statistical tests. And then we have the latest plugin, which is Peacock. So this stands for peak extraction and quality oriented quality control. So this plugin works both with flow and mass cytometry data because it can also handle near zero values very well, and it detects stable density peaks across overlapping beams. So basically, each different algorithm will work slightly different. So there is no best algorithm for quality control. Otherwise, we would have only one and then it would be done. But as you can see here on the example, we have some raw data here. You can see across time of the acquisition, the density, how many events we have across acquisition. We see we have an interruption at the end of acquisition. So, and now you can see when I apply those algorithms, what output I get. So with Flow Clean, I remove this area here. With Flow AI, which tends to be a little more aggressive, we have a significant more events removed from the acquisition. And also with Peacock, we have probably something in the middle. So we have both the end where we have after the interruption and also a part before the interruption. Yeah. So we might choose one or the other algorithm based on if we want to, if we cannot afford having a lot of events removed. Then we would use, Flow Clean in that case, if we study their populations and so forth. But if we want to make sure that we remove all of the unstable events and even at the cost of losing our rare population, then we would use Flow AI. And the next step, so now we have normalized our samples, we have scaled correctly, we perform quality control, and we have our clean samples. The next step is concatenation. So concatenation basically means merging our samples into a single FCS file. Here, for example, I have 4 different donors. So you see 4 different samples. And basically what concatenation will do is to take a subset of our cells. Yeah, so we need to set a number of events, and that should be equal. So that way is actually how we downsample through this concatenation window. So we can set a value of 20,000 cells, and we can take now 20,000 CD127, in that case, cells. Yeah? So now that way we also normalized the number of events. And then what concatenation will do is to generate a new FCS file where we have all of our 4 donors into one FCS file. Yeah. And now we have also a new parameter called sample ID, where we can now identify the 4 different donors that we merged together. In addition, we can also, through this concatenation window, we can add keywords as parameters. So we can select under the advanced options additional parameters, and I can choose here any keywords that I have previously set on my workspace. Yeah. And that's very helpful, as we're going to see later, in comparing the different treatments that we have. For example, here I set a keyword donor ID, and now I can use my concatenated file and put on my axis donor ID, and I can separate between donor 1, 2, 3, and 4. Yeah. And this I can do for as many categorical parameters I have in my experiment. If I have time points, treatments, donors, stimulation statuses, anything I can add here. So then I can later on gate on those and then compare what is the impact of each one of those conditions on my final clusters, dimensionality reduction, and so forth. And now then our next step is, since we have our concatenated file, we can perform dimensionality reduction. So now we have our high dimensional data in a merged concatenated file. Now we have many different populations, so many different subset of cells coming from the different donors and treatments and so forth. So now we want to generate a low dimensional space. So we want to project all of those cell population in a 2-dimensional plot. So basically the way dimensionality reduction works is that each one of those islands, so if we can call this an island, each one of those islands represents a distinct cell phenotype. So cells that are having similar phenotypic expression for all the given markers we select will be close to each other in this 2-dimensional space. So that's a general overview of how dimensionality reduction algorithms would work. And in addition, in Flojo, we can also represent those t-SNEs or dimensionality reduction space with heat map. So I can heat map this embedded space for CD3. So now I can see, okay, those clusters here, those islands are my CD3. Here we have some CD20 cells, and here some monocytes. Yeah. And in addition, I can also, if I want, manually gate, and I can drag and drop and overlay those populations on my t-SNE. So now I can see here in blue color my CD3 positive cells that are in this spot, CD14, so you see its islands close together, and also CD19 over here. Yeah, so that's how you can prove that islands on a Disney mean cells with similar phenotypic expression. Yeah, our B cells would be here, and other cell population would be there. So we do have many different dimensionality reduction methods. So they all tend to do the same thing. So again, group similar cells together in one spot, and different cells would be in another spot. But they do have differences in how they project the data. For example, if you take a look here at B cells and CD4 T cells, yeah, there's pink and yellow populations, you will see that those are far away in the UMAP, TreeMap, EmbedSum, and PathMap. Yeah, so basically in those algorithms, we have a better global structure of the data, meaning that when we have different populations, phenotypically different populations, those will move far away from each other. So if they're two different, will be far away from each other, and if they're closer, phenotypically would be also closer in this 2D space. And that does not happen in Disney, where the distances do not matter. So this white space between my islands here, my B cells and CD4 T cells, you see doesn't play a role. Yeah, they're close to each other. And in fact, we also have a third cluster here of B cells that is even further away from our B cell island here. Yeah, so this is actually this corresponds to the local structure of the data. And of course, this has different uses in our experiments. So for example, if we want to visualize those possibly transitionary phenotypes or T cell exhaustion, monocyte differentiation, and so forth, those options would be better compared to Disney, because we would see the differentiation, and we would see the different phenotypes or trajectories from one phenotype to another. On the other hand, Disney would be really nice to see more resolution if you're searching for a rare cluster. If you have wild type knockout mice and have very slight difference, Disney would be the way to go there. So now we have chosen one of those dimensionality reduction methods, and the next step is to cluster. So we can, dimensionality reduction and clustering go hand in hand. Yeah, so one is not particularly helpful without the other. Yeah, so with clustering, basically we're gonna split those data into phenotypically different population and unbiased approach. Yeah. So imagine now we're after Disney, we're on Disney, we have our Disney map here, and we want to characterize each one of those islands. So now we'd have, we'd have here to gate on each one of those, and manually again characterize what is their phenotype, right? And that would be very time consuming, and we would lose the whole idea of the high dimensional workflow, because now we would do the same thing as manual gating, but just having a fancy 2D plot. So what we want to do is to split ourselves with using one of those algorithms, our data in phenotypically different population. And we can use EXIT, Phenograph, or Flowsum for generating clusters. Yeah, and those clusters then we will find in our workspace. So under our concatenated file, would have a series of cluster or populations, and then we can either overlay those into our Disney or our UMAP and so forth, and see exactly where each one of the phenotypic different populations are embedded in this two-dimensional space. Yeah, so that's why clustering will help us visualize and offer a fresh perspective on our data. Then the next step is Cluster Explorer. So Cluster Explorer is not going to perform new population, or it's not going to do extra analysis, but it's just going to combine whatever we have gathered so far. So basically, we will get with Cluster Explorer a series of 4 different windows here, where we can interact. So I can click on any cluster or any island here on my Disney, and then I will visualize in a line trace plot. I will visualize across the x-axis the expression of the different parameters. So each line is a cluster. I can I can gate here, so I can gate on my CD3, CD45, etc. And I can see which clusters frequency. Yeah, here on the left I can see the plot with the frequency, so I can discover my rare cluster or most abundant clusters. And finally, we have also a heat map view, where again we see the number of the different clusters and the expression of the parameters we selected. So it's a very helpful tool to identify and profile your clusters. So now we are at the comparison point. Yeah, so we have performed clustering, we have performed dimensionality reduction and so forth. So now we need to compare ultimately, because remember we have concatenated file, so we have all of our samples in one. I will show you the manual way first. Okay, you can of course always gate on your differences. So I can gate on my condition 1 and condition 2 and show the UMA in that case, and I can visually see, you know, the differences from condition 1 to 2. So I place the gate here. So here I have an island that is present on condition 1 and slightly reduced on condition 2. If I wanted to do it manually, I could place the gate, overlay the population, and then show histograms. Yeah, time consuming. It is interesting to see where exactly the differences are. So I can see in my histograms, okay, here those are HLA-DR positive, CD38, CCR7 positive, compared to the rest of my sample. So, and then I can move the gate and those histograms would refresh and I would see my what each given island expresses. Yeah? But there is there is a much easier way to do and to very quickly trim down the differences. And this is using the Cluster Explorer heat map window. Yeah. So again, let's say we have 3 conditions, and condition 1 we have absence, and condition 2 as well, and condition 3 or time point 3 we have those 2 different clusters. Yeah. So now what I can do with the heat map window of Cluster Explorer is to add my conditions as nodes, as columns on the heat map. So what will this basically do is to then show me the frequency of each cluster across the different conditions. So it's not anymore in our eyes and how we visualize the Disney or UMAPs, but it's basically the statistics or the percentage of each cluster across the condition. So if I see, for example, here, my clusters 26 to 30 here are have a yellow color. Yeah, so that means they have high frequency on the time point 3. Yeah, and of course I can then export those statistics as frequencies and then use them later on for my analysis. Yeah, and together I can have for each one of those clusters the signature expression and show what exactly are the differences from time point to time point. So then you don't lose time looking at different t-SNEs to check visually where your differences are, but you just take all the information you need automatically in this heat. Yeah. Something else I want to mention is the Euclid plugin. So I know in many times you can use different algorithms to cluster, and this could produce many different results. So if I run phenograph, Lawsome, or Exit, I will get probably 3 different results in terms of how many clusters I have. Yeah? So how do we choose then which clustering is best for our data set? And trying to address that, we have the Euclid plugin. So basically, this plugin will perform 2 statistical tests. The one is the cluster check test, which basically will check each one of our clusters and will detect bimodalities, so bimodal distribution within those clusters across our parameters. So that means if, for example, I have a cluster which is CD4 positive T cells, yeah, and I have within this cluster CCR7 positive and CCR7 negative events, then this test would detect this as bimodal for CCR7, yeah, and that basically will suggest that there is under-clustering, yeah. And the under-clustering is very important because if we have more clusters within a cluster, then we could lose a lot of information in our results. Yeah. So whenever now I would have a very high value, like a yellow value here, for example, cluster 14 for CD19, that would mean that probably this cluster could be 2 clusters. Yeah. And the second test is basically the opposite. So it detects overclustering. So overclustering in general is much easier to detect in a data set, also using Cluster Explorer. But what this test will do is to compare each cluster to each cluster in the high dimensional space. And if those 2 clusters are very close to each other, then it would give us a very low value. So that would indicate that it might be that those 2 clusters are actually one and they should be merged. Yeah, and here you can see on the right, we should be seeing actually mainly high values. That would mean that those are separate clusters, and dark values would mean that we have overclustering. Yeah, so in any case, we can use different clustering methods and then apply the Euclid plugin and see where we get best resolution or best results. Yeah. Okay, and finally, just the last plugin I will show you here is the violin box. So we're, of course, very jealous of the plots that are generating in RNA-seq analysis and so forth. So we also want to have such plots in flow cytometry. Yeah. And so basically this plugin will generate violin box plots or heat maps or correlation plots. So after I have the different kinds of clusters I have created with the algorithms, I can choose my parameters and my clusters, and I can generate those nice plots to export for my presentation or publication. Yeah, so it's just a summary, of our work and see, visualize what are the differences of cytokines or any other parameters I want to study in my cluster set. Okay. Okay, so that's it with the presentation, and I will now skip to Flojo. Go to Flojo and show you how we can do all those things. Okay, okay, I'll check the Q&A box once again and the chat box. Okay, we're good. Again, if you have any question, please let me know. Okay, so I will open here my Flojo, and I will open a workspace here that I have, and I will explain what we're seeing here. So I have a group here called stain samples. Yeah, so I have 3 samples, and these were PBMCs that were stained with 27 colors. So it's a multi-dimensional flow experiment. And here basically it was a panel for general immunophenotyping of different populations. So we would see T cells, B cells, monocytes, and every kind of immune cell. So how do we start here if we want to analyze those data? So very first thing is to create some keyword. So I can press right click here and add the keyword. Yeah. I have a keyword here called donor ID. Yeah, so I can select here and I can add the value. So I can add here 1, 2, and 3. So those are 3 different donors. And now I have this keyword that I can use later on for my comparison of the 3 different donors. Yeah. Now, typically the next step is to perform quality control. Since those samples were acquired in the same day, there is no need for normalization in that case, but we still need to perform quality control. So for quality control, I'll use one of my plugins. So initially you can detect if you have bad quality of your samples. If you go to Tools and select here, check sample quality, this will give you here on the circle a color. If you see any purple or yellow color, like here, that means that you need to have quality control. There was something problematic on those samples, and that's the case for us here. If you see green or blue color, then you're okay. You probably don't need quality control, but in that case, we do need. And now we'll use under my workspace and plugins, I will use my Flow AI plugin. Yeah. So for Flow AI, it's important to remember that we will use all of our uncompensated parameters. Yeah, so here I select all of my uncompensated parameters, and below there are some tunables, so you can select a combination of statistical tests if you want. I would start with all checks here. And there are some other tunables to make the algorithm more or less aggressive. We have set here a default that would work in most of the cases. But if you want to, adjust those values, then you can also, read the paper here below in case you need to know how exactly to adjust those values. And then I can also select here, remove outliers. So this will also remove spikes that we have in our data. And finally, you can also save the R script. So this R script will help us in case you have any issues with running Flow AI, you can save this R script and send it to us to see what exactly is the problem. And then I will simply press OK. And this will take a while, depending on how many events you have within the sample. So now I run it only for my first sample, and then I will run it, of course, for my other two samples here. So here I had 742,000 events, so that's why it takes a bit longer. Yeah, we always need to run it on the full sample, just to say also this, that we cannot run flow AI in a subpopulation. We need the whole sample to run. And that was it. So now flow AI is done. And we have two population bad events. So 23% of my cells were removed, and I have 76% that were kept. So if I want to visualize those results and the plots that Flow AI generated, I can use my folder of analysis. Yeah, don't forget that whenever you run a plugin or anything on your dataset, in this folder that you save your analysis, everything will be also saved. Yeah, so if you go now to Flow AI, you can have a picture here of where exactly and how your events were removed. Yeah. So what we see here, those events were not removed because of the flow rate. So those events were removed because of the signal. Yeah. So the median intensity values here, I don't know if it's very visible, but we have a decrease on the signal here, and that could create artifacts. So that's why the algorithm removed this area here. Jan has a question. Can you run Flow AI on multiple samples in parallel in one workspace? Yes, we can do that. And here I only have three samples, but you can drag and drop this Flow AI node. So after you finish one sample, you can drag and drop this to your group. Yeah. So basically now, this will start calculating for the rest of my samples. Yeah. So so that's the way to do it. Otherwise, yeah, actually that's the only way of doing it. Because if you first select all of your samples, if you multiple select your samples and then run Flow AI, it will open the Flow AI window 3 times or 10 times. So I wouldn't do that. I would first run it in one sample and then drag and drop the Flow AI node to the rest, to my group or to rest of the sample. So now we're calculating for the rest samples. Okay, my third sample here is done. It also removed 70%. And now we're just waiting for the second sample here. In the meantime, if you have questions here, just type it in the Q&A box. And then after my second sample is going to be done, then I will delete this Flow AI node, yeah, because this Flow AI node is basically whenever you change something or you start if you start changing gates and so forth, this will recalculate. Yeah. So by deleting it, remember you don't delete your analysis, so you don't need to be worried here. Your flow AI would still be there, but you just avoid recalculating in case you change your gates or change something. Yeah. So I really suggest deleting this node entirely, and then you see you have good and bad events here. Yeah. Federica has a question. Do you suggest to perform Flow AI also for 15 colors, so less than 27, even if performed manually? Absolutely. Yeah, absolutely. Even if you have much less colors, not just 15. So the quality control plugins are really not having to do with the high dimensional space. So if you have bad events within your samples, I would nevertheless perform quality control. Yeah. So because also with manual gating, you can create artifacts. Yeah. So ideally, we would want to remove also those events. Yeah. So absolutely. Okay. So now we have our good events here. We have our good events here, and we need to we need to select our good events. So I will select my good events, and typically I like to export. I like to export my events, my good events into different cell populations. Yeah. So with Flojo we can actually use the file here, file tab, and press export concatenate populations. So here in this window, under the export tab, you can select here include all. So we will include all of the events, and below I can just have some advanced options, and I can select a name for my file, so I can use as export, or I can put something like QC here. QC. And then I can select export. Yeah, so this will export to 3 FCS files, and then I can use those for further analysis. So this way is better than using this gate and so forth. Yeah. So I'll press export. And then I will select if I want to have them in the existing workspace or in a new workspace if I want. Yeah, so I will select here existing workspace, close, and now I have my quality checked samples within my workspace. Yeah, so now I have only my documents. Now, now I can add those samples into a new group. So I typically create a new group like QC Flow AI, and I can drag and drop those samples within my group. Yeah. And so here, I do not, I have some gates here. So the next step, now that we have our clean samples, is to check for, to place some gates here. Yeah, so remember we have now all of our whole sample, but we don't need, for example, granulocytes, we don't need leftover erythrocytes or whatever. So we need to place some gates. Yeah, so I place a gate here for my lymphocytic and my monocytic populations. And then I placed, I think, another gate, which was my CD45 positive cells. Yeah, so I can, I had a gate here, side scatter versus CD45, and I included this population here. Yeah. And now I will select, so we need to reach our analysis till the population we want. So I just reached it till my CD45 positive cells. And now it's the step of our concatenation. Yeah, so I can select my CD45 populations, and I can go to my export concatenate and concatenate populations. Yeah. So under this window, in the concatenation window, I can normalize the number of events here. So I can either include all events, or I can normalize to a specified number. Yeah. So I can, this is also known as downsampling. So now I can take, for example, 100,000 cells, and this will take 100,000 events from each CD45 positive gate from our samples. Yeah, so that way we have equal number of events across the donors. And additionally, as we said before, we can use additional parameters, and here select our keywords. Yeah, so I will select now my donor ID keyword here, and I can press OK. And then you will see that my keyword will be added as a new parameter on my concatenated file. And then I will press concatenate, and then I will have a new FCS file. Yeah, so here I have it already processed. And so that's my concatenated file. I have 300,000 events. Yeah, and I can open this file, and I can use any of my keywords. I can use my donor ID, for example, and I can see indeed that here I have 3 different donors. Yeah. I see we have some questions here. Is Flow AI affected by parameter scaling, or does it take the scale from the flow cytometer that is saved in the FCS file? No. No. So the parameter scaling here I think it will not affect our it will not affect these algorithms for Flow AI. Another question is, sorry, is this traditional flow or is it spectral? So no, this is traditional. Yeah, so this is not spectral. But in any case, this should not be an issue with this workflow. And then at the stage of the concatenation, shall we include only viable single cells, doublets removed at this stage, or it does not matter? So yeah, so we should exclude the dead cells. We should exclude the doublets and any population. So we're just gonna place our normal gates, like for example, till if you have a dump channel, you would also remove those. Yeah. So basically, you will remove any population that you don't want to see on your Disney or on your uh, uh, clusters. Yeah, so you will just read, if you would want to see only your CD3 positive cells, then you reach it till your CD3s and just focus on them. So it's up to you, but normally you would avoid having all of the dead cells, doublets, and so forth. Yeah. Another question we have is, you perform all the steps concatenation on uncompensated parameters. Why? Yeah. So maybe I can show you again here, just for an example. In the concatenation window here, in the parameters, you have three options. So you have all uncompensated parameters, you have all compensated parameters, and custom set of parameters. The rule here is that when you have the same compensation matrix between the files you're concatenated, then you will use the uncompensated parameters, yeah, because then we can apply the matrix that we have already in the new FCS file. Yeah, so same compensation matrix, then we use uncompensated parameters. When now we have different compensation between the samples, yeah, so if I have 3 time points and 3 different compensations, then I have 2 options basically in that case. If I select all uncompensated parameters, it will create a new FCS file that I can put any of the 3 kinds of compensation that I have. If I select compensated parameters, yeah, so each time point, each file will have its own compensation. So the compensation will not be the same. And in that case, the concatenated file will have the compensation matrix integrated in the parameters, and it will be named FG-comp, FlowJo-comp. And there you cannot change the compensation anymore. So it's you cannot apply any new matrix. Yeah. So it's up to you which one you would choose, but typically when we have the same matrix, we use the uncompensated parameters. Yeah. Okay, so let's move forward here. Where were we? Here. So we have 3 donors. Yeah, so remember we have a keyword here called donor ID. So I can use this keyword to place gates, and those gates will be super useful later on for our comparisons. With the histogram, you can make a quick gate by pressing right click and create gates on peaks. Yeah, so now directly, very easy. I have 3 gates here. I have my donor 1, donor 2, and donor 3. Yeah. And its equal percentage is 33% for each. Yeah. So so now the next step here would be the dimensionality reduction. Yeah. So I can use many different algorithms. So you can find under workspace, you can find Disney. And Disney is implemented in FlowJo. And then we have other dimensionality reduction algorithms under plugins. So here you will find, for example, we have UMAP, we have TreeMap, PackMap, and fate algorithm. So we have many different to choose from. Yeah. Now I will select an algorithm. So it was mentioned a lot, the performance. Yeah. So running out of memory from Jan, etc. So we will use an algorithm that behaves very, it's very fast with creating dimensionality reduction space. And that's the embed sum algorithm. Yeah, so this you can perform in a very quick way an embedded space. Yeah, so this is a plugin, and you will find it here under plugins and embed sum. Yeah, because it has an initialization step with self-organizing maps, such as Flowsum does. It will perform much faster and can handle larger data sets much faster. Yeah, in cases that it would take you, for example, 1 hour or more for a Disney or a UMAP, and Betsum will make it much faster. Yeah. So for Embedsum, here we will select our compensated parameters. So I have my compensated parameters included. And here I will deselect any of the gates that I have gated in or out. For example, remember I have already gated on CD45, so I will exclude the CD45 here. Yeah. And again, if you have any other viability dies or dump channels that you already gated out, you would exclude those because they're not going to offer any better resolution on your embedded space. Yeah. And the Embedsum has different landmark layouts that it can produce. So you can use either a Flowsum map that you have already and input it. You can use a UMAP layout, Disney, or the classic way that is the Embedsum. Yeah. There is also the option to perform supervised embedding in UMAP if you want, and you can add, for example, either clustering results or any keywords or treatments you have. Yeah. And now I'll press OK here. I'll leave the defaults that in most of the cases work well, and I'll press OK. So Embedsum now is calculating. So we have 300,000 events. This would take a significant time for Disney and UMAP. Let's see how fast it does it here. So it's done already. It took less than a minute, so it's very fast, and it created a nice two-dimensional space. So now we're visualizing all of our data. I can check the space if I want. I can select my color map axis, and I can use any of my different parameters. For example, I can see, okay, where are my monocytes? Here are my monocytes. I can check my CD3 positive T cells. Okay, so all those islands here are CD3, and you know, I can go further and further in my with this heat map and see what each spot corresponds to. Yeah. So if I want to do it on the layout editor, I can also, you know, drag and drop my sample, or I can just press right click and copy to layout editor. And the manual way of comparing would typically be to overlay here my different gates for my donors. Yeah, for example, I can drag and drop my donor IDs here, so my 3 different donors, and I can drop them onto my embed sum. Yeah, so now in each different color, I see each one of the different donors. Yeah, if I want, I can also change the view here. I can use maybe a counterplot view. Yeah, so now I can see a bit better, you know, the differences. For example, this cluster, this island here seems to be present in donor one. For some, we have equal contribution here, and we do have some differences here and so forth. So that's the manual way of comparing, you know, the results from donor to donor. Yeah. So now we have our embed some space here. We can, you know, you can create multiple color maps. So I can select my compensated parameters here and, you know, make all those nice plots where we can see what each island correspond to. Yeah, so they're all CD45 positive. We can see CD64, CD4 T cells, monocytes, and different subsets. But again, we don't know exactly how many clusters we have, and we want to avoid gating on each one of those islands. Yeah, and this is why we're gonna use clustering. Yeah. When you have a data set where you're not sure how many clusters you should expect, then I would use one of the algorithms which are phenograph or EXIF. Yeah, those will automatically identify how many clusters you have. Yeah. If you know more your data set, if you know how many clusters you should expect approximately, then you can also use FlowSum, because FlowSum will require you to input the number of meta clusters that you have in the data set. Yeah, so it's sometimes hard to know exactly how many clusters we have. So now, in order to perform clustering, you can go to plugins, and here I have used before Phenograph. Yeah. So if I select here plugins and go to Phenograph here, I would need to select all of my compensated parameters apart from the ones we gated in or out. Yeah. For example, here I will deselect CD45. Yeah, so I will then leave this K number, so the number of nearest neighbors at 30, and then I can simply press OK and run the algorithm. Yeah. I see a question. Hello, when I run Phenograph, I have only R and Excel files, but no PDF in the output folder, and I do not have Phenograph gates in the workspace. Could you help me understand why? Okay, so I will cancel here. So when you run Phenograph, you shouldn't have any PDF. So if I go here, I previously ran my Phenograph. Yeah, you will only have R files and CSV files. Yeah, so it's not expected that you will have a PDF here with a different of clusters. So if for some reason you cannot see your gates in the workspace, then you can take and reconnect those by pressing here. You can press on your sample, right click, and select import parameters from CSV file. Yeah, and then you would need to input the CSV file that you have from running Phenograph, and that would reconnect, and then will place the gates, the phenograph gates on your sample. Yeah. Okay. There's another question here. What is the K value in phenograph? Yeah. So basically, the K value is the number of nearest neighbors that will be used in order to partition the data in cluster and compute the phenotypic similarities. Yeah. What does this mean? So a K value of 30 is great to start with and covers a lot of different data sets. So if you decrease this number, then you will have less neighbors included, meaning we will study less cells in a specific region, and that will give you as an output more clusters. Yeah, it will tend to over cluster if you reduce this value. If you increase the number of K nearest neighbors, then the opposite effect will happen. So you will have less clusters. Yeah, but a value of 30 it should be good to begin and check how many clusters you have in the data. Okay. So now with phenograph here and the K value of 30, I have identified 37 different clusters here. Yeah. If I want to check those clusters and what if my clustering was good or bad, again we can use the Euclid plugin, yeah, where we need to select our parameters. So I can select my parameters, and I can check for under clustering and over clustering, and I can select my phenograph output here. Yeah, and then I press OK. And basically, this will show me how good my clustering was in that case. So if I choose to play around with different K values, I can run, for the different K values, Euclid, and see if I, if I tend to overcluster or undercluster. Okay, and that was it with Euclid. So on the left, we see the under clustering, which is more important. And in general, we'll see very low values. Yeah, so the high value should be the yellow. So if you see a lot of yellow here, would mean that you have under cluster, and you should probably change your K value or your clustering algorithm. Yeah, so here we're good. We only see at cluster 21 that we have bimodality for CD64, CD8 in that case. Yeah, and later on we could check that with Cluster Explorer and see if we, have under cluster. And on the right we can see the score. So again, we have a good clustering result, have very low. Very low similarities on the comparisons between the clusters. And we can also check that with Cluster Explorer. For example, now that we have this table, and if we found out our specific, cluster of interest, then we can compare and see, are we sure that this cluster is one, or could it be that it's the same with another cluster? So that's the way to go. Okay. Okay, and speaking of Cluster Explorer, now we can jump into it, and I can select here under my plugins, I can select Cluster Explorer. So, so here in the Cluster Explorer window, we will select our phenograph clusters. Then the parameters we want to visualize. So again, I will exclude my CD45 parameter, and then I can select my embedded spaces. Yeah, I can use here in that case both and see how they look like. Yeah, and I'll press OK. And this will quickly generate the 4 windows we were mentioning before. I'll just try to expand those so that you can see them better. So this is the line trace plot. So it's very useful to visualize what each cluster expresses. Then we have our heat map here. Then the cluster frequency here, and somewhere hidden back there are my dimensionality and my embedded spaces of Embedsum and Disney. Yeah, just organize them a bit nicer here. So now we're visualizing the concatenated file. What can I do here is you can interact with every component. Yeah. So first of all, I can use my Disney or Embedsum, and I can start clicking here, and then I can see this island here, which is actually here in Disney, has this phenotypic expression. Those are some running, those are some NK cells, and I can multiple select as well. So I can select this cluster here and this one as well, and I can visualize their differences. Yeah. So you see now those were separated into 2 different islands. Yeah. So 2 different clusters, but they're very close to each other. So if we would suspect that this could be one cluster, then we can check this signature profiling. Yeah, they do seem to be very close phenotypically. Those 2 lines are overlapping, but we do have differences. So we do have differences for CD27. We do have differences in CD38 and CXCR3. Yeah. So that's exactly, again, a proof of dimensionality reduction, and that it brings similar cell population together, but also a proof that our clustering was good, because they are different phenotypes, and that's why they were separated in different clusters. Yeah. Okay, what else can we do? So we have a question here. Running Cluster Explorer on M2 Mac frequently crashes on flows. Any hint? This should be this should be a technical support question. I cannot say why it would crash here, but you can contact, in that case, flojo@bd.com. So maybe I can type it here, the email address. Let me increase the text. The email should be this, @bd.com. Okay, so if you have any text support question, you can send an email here, and they will help you with Cluster Explorer. Yeah. Okay. Let's go back to Cluster Explorer. So in addition, we can gate here, which is also very helpful. So I can place a gate here on my CD3 positive T cells. Then I can find, for example, where are my CD4s? So those are my CD4s. And then, you can place as many gates as you want. For example, you can go for CCR7 naive T cells and so forth and so forth. Yeah. I will delete now those gates. Okay. And the same with the heat map. Yeah, I can change the visualization as I want. Can change the colors. And all those plots, of course, can be exported. Yeah, you can copy the content and you can show it. You can show, okay, this is cluster one, this is its frequency, and you can add those plots on your presentation or publication. So now we focused a lot, on discovering the different clusters. But what's important here is to actually compare. Yeah. So we see all of the clusters in all of the samples here, right? So if I want to compare, I can do it with two different ways. So first of all, I can either compare by selecting the global node and changing it. So instead of now the concatenated file, I can use my donor one. Okay, that's the view of donor one, donor two, and donor three. Yeah, so slowly but surely we see some differences here. Yeah, so you can either visualize here, for example, I don't know, I just picked a random cluster here, which I want to see for the three different donors. Yeah, so that's indeed more abundant in condition in the donor number 3, sorry. Yeah, and those are some CD8 positive, some CD, yeah, don't know what exactly, but CD8 T cells. Yeah. So that's one way to do it visually, yeah, and all those plots will refresh. But the favorite way, and this is what we're waiting for, is actually to use the heat map window, so that you don't have to manually check one by one. I can do the following. I can select here the option button and select add nodes as columns. Yeah? So now in this window, I can select my gates that I have from my workspace. Yeah? And I press select new columns, and that's it. So now I can see for each one of my clusters the frequency across the donors. Yeah. So now when I see high values like this one here, so this cluster here is 77.77% coming from donor 2. Yeah, so it's absent or less prevalent, so to say, from the rest of the donors, but it's present on my donor 2. And that way I can multiple select all of my differences. Yeah, so I can check here. This one not so much. Yeah, so I can trim it down really to the clusters that are significantly different. Yeah, there's a lot there. Yeah. Yeah. So all those clusters are different from donor to donor. So you want this information, now you can press options and copy content. Yeah. So I can copy this content and I can paste it to my Excel. So I can open an Excel sheet. Yeah. Okay, now it looks a bit busy here, but if I can make it much better, can press text to columns, because it shows all the values of the heat map in that case. Yeah. Comma separation. Okay, I'll finish it. Okay, so here are all my values of the heat map, and what I want to see and focus on are those guys here. Yeah, so those are percentages of each one of my clusters. Yeah, it's one of my 30 something, 38 clusters across the different donors. Yeah, and I can use that and make nice plots and see what is the significance of those clusters. Yeah, and together with the signature profiling, we can show this is my cluster, this is its expression, and this is the frequency among our treatments or donors. Okay. Cancel this. Okay. So that's an overview of how Cluster Explorer works. So you can really do a lot. You can spend a lot of time in Cluster Explorer, but you really dive deep into your data set. And just to finish here, because we went over time. So if you want to generate plots, as we said before, you can go to plugins and use the Violin Box plugin. It's also part of the plugin play bundle, so you can download it and use it easily. And again, always we need to select parameters we want to see. So we'll select some of the parameters and categorical parameter, which will be our phenograph clusters, and then press interactive, and this will generate and will lead you to a web page where you can change, you can process and generate different kinds of plots. Here we are. So first we have an MFI table for each one of our clusters. So it's nice and easy that you can just copy this also if you want, statistical comparison. Then I can switch to heat map. I can make a heat map for the parameters that we have selected here. Yeah, so that's my heat map for all of my different clusters that I have. I can change the scaling. So if the scaling is by row or by column, you can also change it here in case you have a general scaling. So now you can see much better. You can add a bubble heat map if you want to visualize it in a different way. And then apart from the heat maps, you can make Violin plots and box plots together if you want. So that's the step where we can define what each cluster and just extract the information of our clusters. And you can, in addition, you can adjust the plots if you can use the options and you can adjust the size and the fonts, the size of the fonts. Also, here in the violin box, you can flip the violins, use a dark theme overlay, have some statistical comparison, and you can also always download those plots for your presentation. It took a bit longer than expected here, but I do have faith on it. Yeah, I'll just keep it running. Okay, so if you have any more questions, just you can add them in the Q&A box. Otherwise, this is not going to finish, I don't know. Yeah. Okay. So thank you all very much. And again, if you have any more questions, you can contact here, or I will also write you my email address in case you have any issues or you're getting started with a plugin and you need some more support. Please, I will be happy to help. Okay. Okay, thank you very much and have a great day. There is a question. Can we have access to the video? Of course. And this should be, I'll show you under Learn and Webinars. And if you go now at the bottom, very bottom, it's here under Recorded Webinars. And it should be here by tomorrow, I guess, or I don't know, today. So in the worst case, next week, it will be here now as the latest one. Yeah. Okay, here are our plots. So just saying goodbye with those plots here. Thank you very much all and have a great day.